rest of the components of the signaling cascade. So also, once we have our network visualized, then we can play around, but I want to show you cluster-defined network, and then it will color the nodes based on the sub pathways inside that the high-level signaling pathway that we had plotted or that we have represented as network. So if we go over here, if we click, sorry, it's a little bit slow, so is everyone following, or?
I think so, Juliet. Even if they're not following, I just put in the chat that they should just watch you how you did the screen, and then they can follow along. Thais is asking, how do you put the colors? Can you show again this last step, please?
So when we do right click, and then we go to Reactomify, then there is this option that says cluster-defined network. So when you click on that, it shows you, it's just color based on the sub pathways that we found here. So when we do click on them, they will show you that. So if you see, if I unclick, then they have the same color. And so you can see, this is what I mean, for instance, in this case, when we have the acetylcholine regulates insulin secretion, there are many interactions over here, like the G proteins subunits, but the network is just showing us like these two. From all of the reactions that we found inside this pathway, is just showing us two of them. So there is some difference. There are some differences between what we can see on the over here, even over here, and what the network displays, because of the way it was constructed. If we recall that, like integrating different biological databases and using Reactome to test the model. So if we see over here, for instance, sorry. So we can move around, and then it will color in yellow all of the nodes that are part of that specific pathway. And then over here, so once we are in Cytoscape, then we can also use the other features that allowed us to use a little bit of network analysis, but I think that that's a topic you will cover deeply in a different section. So let's say just focus on the display. So also, if you see over here, we can also access disease, but only infection diseases. And it was actually a request that I did to the software developer that maintains the app. I don't fully understand why the other disease can't be uploaded to this particular plugin, but definitely he said that it was possible to upload the infection diseases, and we find over here the one in which I work on. So I'm going to continue with the presentation, and then I want to show you some of the networks I can create with the components in this signaling pathway. So let's continue.
While you go back to the presentation, Paula has asked in the chat if there's also something similar for bacteria species.
For bacteria species, I haven't seen, because this is specific for Reactome. Do you mean if there is like another plugin in Cytoscape for that?
I think it's it's what she's asking. I think so, yes.
Yeah, I really don't know.
Hi, sorry. Yes, I'm asking because I work in bacteria. Sorry, I work in bacteria and Staphylococcus aureus and Pseudomonas aeruginosa. And here I only see mus musculus and human. So I don't know if you know something for this, because the graph are very nice and everything, but no, very specific.
Yeah, but Paulo, I think that there is some sections that can cover that, right? I personally don't know, because I work more in the human side of the host parasite interaction. Yeah.
Okay,
yes,
thank you.
And we're gonna have, Paula, we're gonna have a talk on, I think it's Wednesday, when the people from the CARP group, which has developed EcoPsych and BioPsych in the whole site range of databases, they're really focused on bacterial species. Their main focus has been bacterial. So there you can always construct, for instance, a metabolic network and display using a graph using cytoscape. So I think it's going to be covered on the upcoming sessions.
Okay, thank you very much.
Thanks for the question.
Yeah. And so, as Henning also mentioned, we have the component of diseases in Reacton. And over here, we find the one that I started, because I hope that it can continue growing. So since we are going to use some transcriptome that comes from the infected macrophages, I'm going to give you a little bit of a review of what Leishmaniasis is, and so we can have a little bit of the context. So we know that some of us, we know that Leishmania parasites are intracellular, and that the main host cell for them are the macrophages. There are different Leishmania species causing different clinical manifestations, such as the cutaneous, mucosal, and visceral Leishmaniasis, but yet all of them reside in the same cell type, the macrophage, the cell that is in charge of their elimination. So normally, when we take the transcriptome data of infected macrophages, we would like to compare, as in many other infection models, infected cells versus uninfected, or to follow over time the dynamics of the transcriptome. And for all of those cases, of course, we start with a list of differentially expressed genes. And what we want is going from that to a list of biological processes, but it's also important the molecular interactions. That is why we are here to display the networks, because actually by knowing how those interactions are occurring under the context of biological responses is a nice pipeline to identify molecular targets. That is basically what, at the end of the day, many of us want. But often, what do we get? Often, at least in my experience, with my own data, by reading papers, by looking at the data people show in different conferences, often we get unlikely biological processes, like neural system, where we are talking about a system of a macrophage, muscle construction, or things like that. That doesn't make much sense. Or really high level biological processes that they are kind of expected, right? They are the ones with the best statistics, that is the FTR. The smallest FTR are normally the ones that the big biological processes, or the high level pathways, as it's called in Reacton. Or if we have some specific biological processes, like we can identify, for instance, the messengers of the specific cell response, like the arachidonic acid metabolism. Oftentimes, we get these biological processes disconnected among each other, because normally what we get is a list of biological processes and range based on the FDR, the statistics associated to the enrichment analysis or the overrepresentation analysis that we have done with our omics data. So for my internship at the ABI, I wanted to address this issue that was really bothering me during the analysis of my data. So I proposed let's build contextualized labels for Leishmaniasis. How do we address this issue of unlikely biological processes? By creating contextualized vocabulary, contextualized labels or ontologies. And how do I address, how did I address this issue of having a disconnected, high level, lower levels of signaling pathways from the overrepresentation analysis outcome by performing clustering of the results, clustering of the pathways based on the shared genes. Because, and this is something that I really liked about Reactome, the fact that they are since they are a hierarchical structure, then we know that if we have a big high level biological process, all of the ones that are below that, then of course they will have those genes in common, if it makes sense. A high level biological pathway will contain all of the genes of the subpathways downstreams. So if we cluster them by the shared genes, then we can get together the high level with the specific biological processes and maybe get better insights of what is actually going on in our data. So with this in mind, and really with the having in mind the classical structure of signaling pathways, I propose the structure for this repository of pathways of interest in leishmaniasis. I call it like that, repository of pathways, but for Reactome paradigm, that is also called a pathway, Leishmania infection pathways. But for me, this is just like the label of a repository within which I will introduce actually signaling pathways that can be important for us to understand our data in the context of pathology and healing or non-healing responses. So I started with this high-level path, cellular responses actually, like or processes, parasite infection, and everything that is all the signaling pathways that are occurring during the first contact between the parasite and the macrophage are going to be here. Phagocytosis, the pathway I started with, and later on I will add TLR receptor interactions and everything that is occurring in the first contact will be within the parasite infection. Then killing mechanisms, the same as I said, all of the pathways that end up in an effector molecule, like the production of ROS, or an effector molecule that will cause damage to the parasite, like for instance, pro-inflammatory mediators. And then specifically a category for cell recruitment, because it's also important to understand what type of cells, for instance, are being recruited to the, or at least what kind of, in vitro models, of course, we will know like what kind of cells potentially are being recruited based on the chemokines and the pro-inflammatory mediators being secreted in infected macrophages. And in Leishmania parasite growth and survival, I'm collecting all of those pathways that actually benefit the parasite. And right now, everything is mainly human right now, but it has deposit. We have already some components of the parasite, and the idea is that the pathway is flexible enough to also curate the mechanism, the SAC mechanism that the parasite is using to hijack the cellular machinery. So this is the structure of the pathway. I'm sorry, this is like the structure. I really did think about that, a classical structure of a signaling pathway. So all the pathways here, it starts with a membrane protein, and all of them finishes with an effector molecule. Being, so let's say in Leishmania phagocytosis, inside this category, I curated the phagocytosis mediated by FCG gamma receptors, FCG3 gamma receptor, and the idea is to add the phagocytosis done with other receptors like the complement or mannose receptor, because we know that when parasite use different phagocytic receptors, the there are different functional consequences in the phagocytic cell. So the idea is to add those other kind of types of phagocytosis mediated by different receptors. And as you can see over here, all of them starts with a membrane protein and finalizes with a specific effector molecule. And so you see, for instance, that this is the anti-inflammatory response. And one person that is querying the database will immediately understand, will immediately get, for instance, the mechanism by which that anti-inflammatory response favoring the Leishmania parasite infection is occurring, right? Not just the general, it might not get just the general like biological processes, like something that is favoring the Leishmania parasite, but how? Like what specific pathway associated to that biological process. So as Kenin showed you, these kind of diagrams are actually built manually. We manually have to create each of these entities. And of course, as you already know, each of these interactions has to be backed up for the original papers in which those molecular interactions were proven. And over here, it's just to show you that there are different pathways getting integrated in order to give that overall or high level biological mechanism that is anti-inflammation favoring the parasite survival. So we will use in the R session, we will use data from this work. That is a dual transcriptomic analysis of macrophages infected with Leishmania major. And idea proof of concept to show that how by having contextualized labels, we can like speed up the process of getting mechanistic insights from omics data by using that data that was already published and comparing with the results that the same authors discuss in their paper. And basically, they didn't realize that they have activated this signaling pathway that is with this GPCR receptor ADORAP2B, with this adenosine receptor ADORAP2B. And this pathway, I selected it because previous literature showed that for other kind of species, that is Leishmania amazonensis, this pathway can be manipulated in order to control the parasite load and at the same time controlling the pathological pro-inflammatory response. And I chose it because I curated this because of that reason and because I was also finding components of the signaling pathways in my own transcriptome data that was from a different species that is Leishmania panamensis. And then I used this data that is a different species that is Leishmania major, showing something that they didn't actually realize in their when they did the enrichment analysis, because the pathway was not found in any biological database. And we can see from the beginning, from the interaction with the adenosine molecule and to the production of the effector molecules that are contributing to, that are benefiting the parasite survival. So in this way, we can use Reactome database to actually compare, to help us to compare, for instance, what pathways are curated among the between the infection with different strains, different species, or not different species or different taxa of a particular parasite bacteria that we are studying in the interaction with the human host. And so next, and this is kind of a little bit of like, if you find this somehow interested, like it might be nice also if you get inspired to create the signaling pathway, the repository of pathways for the pathogen of your interest, because it's actually useful to identify, conserve, or species-specific pathways. For instance, if we say, okay, this has been shown in different species, except or no, the way around. This pathway hasn't been shown in any other species, but just to the response to this particular species, for instance. And that kind of information is very useful when it comes to develop, for instance, vaccines that are targeting some particular cellular response, like is the case for Leishmania. And since we are starting with membrane proteins, if you decide, for instance, to do your own signaling pathway repository, I think that is useful to do it in this way, in which we start with a membrane protein, since they are the main targets at the end of the day. They are the easiest targets. And by including the effectors molecules at the end, then we can know what readouts to expect when we go to the lab and to test, for instance, the effect of targeting, shutting down membrane protein. We will know what exactly to expect to measure as a readout. That will be the effector molecule. And then I just wrap up. I like also visualizing reactome enrichment pathway outcome by clustering the pathways based on the shared genes. I do this in R. I didn't bring the code. I don't know if it can be useful for you as well. It's just that it's easier for me to see, instead of seeing a list of biological pathways, having just like clusters of them. So for me, it was easier to see, like, identify the cluster of lipid metabolism and the kind of lipid metabolism, like the ROS production, bioenergetics, the inflammatory like processes, TCA cycle, and so on. And this way was how it helped me to build a reconstruction of the immunometabolism of affected macrophages. And just all of these pathways has to keep growing. It's not a finished process. And I don't know how long do I have? How much do I have, Pablo?
I'm not sure if Dario knows the time. We started 11:20, so we have 1 hour here, and then the idea. Well, still we have the afternoon is yours, so yeah. You can either choose to. If we start in 1 hour, we can resume from the point where we're at right now, and then go a little bit further on the afternoon. It's up to you, I think.
Yeah, so I would like to then finish this with cytoscape.
Okay.
And then, so if we see the let's say the anti-inflammatory diagram that I showed you, let me see. So over here. So you can see that we actually can use reactome pathway to create the components also of our parasite of interest, and then use all the features that this platform has, that this platform offers. So in this case, all what is from Leishmania parasites is depicted like with a red perimeter. And then when I come to here, it's a little bit slow, my computer. Then we can convert this to the FI network. It takes some time.
In the meantime, Juliet, is this plugin made by the people from Reactome directly?
Yes, from the people in Canada, actually, specifically. Yeah.
Okay.
So yes, so we can see here. Oh, okay. In this case, I have pre-selected when we do the first time, when we use this option of cluster-defined network, the first time, it kind of replicates in the following networks that we build. Because at the beginning, if I open this without having used previously that option, we will see just like undirected arrows, like undirected edges. And then when we run this option over here, cluster-defined network, then is when we get a little bit of more information of, for instance, this over here. This can be either a complex, or this can be either a defined set, which means that they will be members of the same family. So that kind of information we can also explore it over here. I'm sorry, it's a little bit slow when I'm moving it around. Okay, wait. Layout. Then we can use. And we can, of course, use the Cytoscape options to lay out the networks wherever we want. I just wanted to show you that by displaying the networks like this, then it was easier for me to identify the selected pathways that I the selected interactions that I next converted into a mathematical model. And at that point, I didn't use the interface with biomodel, so I basically did it kind of manually. But once we are over here, then we can transform this into a logic model that then we can upload to biomodels and to add the information of parameters or kinetics that if we have it, of course. Yeah. I wanted to, okay. So yeah, basically, I think that you will see with in other sections more details about how to analyze networks. And let's say that for this one, it's just basically the different ways we can visualize them from pathways to networks. And this is basically, I think, what I wanted to show you with Cytoscape. There is also, of course, the option of doing enrichment analysis as well. And if you do enrichment analysis, then we can have the nodules, the nodes color based on the pathways that were enriched, the different pathways that were enriched, basically. Yes, and I think that's for now.
Okay, we have some questions in the chat. So Masoon just asked us if we can export all the information associated to the network to a CSV file or JSON to be manipulated later by any programming language.
Yes, yes, we can. So here, for instance, we have the information of how and we can also do actually enrichment analysis over here to see all the information that we can export when we do the enrichment analysis over here comparing to when we do it in the web page. We can do it after the break, actually. Yeah.
Okay, great. Yes, I think those were the questions. So Juliette, I'm really happy that you brought us an example of Leishmaniasis, which is a tropical, very neglected disease to this Cabana course, where we enroll in some Latin America country, and most of these countries are affected by Leishmaniasis. So I'm very, very happy that you brought us an example that will be hard to see in any other workshop other than this Cabana. So, and in fact, I was just seeing the live doc that we have where people are presenting themselves and putting up their research interests. And I see that there are some people working with Leishmaniasis as well.
Yeah?
Yes, so I saw the name of Fabiano Pais, and I also saw the name of Franklin Calderon, which are trainees here in the course, and perhaps there are other people as well. Well, myself, I'm working with, in a group of Leishmaniasis. We have some publications going and doing some genomics of trypanosomatic parasites.
Yeah.
So I'm really happy that you brought us this very important example of a neglected disease that is very underrepresented in databases. So doing all this effort to enrich reactome with these classic signaling pathways and the immune response associated with this disease is really great. So thank you.
Thank you.
So if we don't have any other question, I suggest that we make our lunch break and then resume in 1 hour at 1:30 p.m. with following you, Juliet, again with the rest of your session.
Perfect. Thank you.
Thank you.
Have a nice lunch.
See you guys in 1 hour.